chapter 16, we are going to learn about amino acids and proteins. Okay, by the end of chapter 16, you should be able to identify the structural features of amino acids. Each amino acid has an alpha carbon, and that alpha carbon is attached to one carboxyl group, one amino group, one hydrogen, and one R group. And there are 20 different amino acids, and each one has its own individual R group. Okay, you should be able to describe the acid-base properties of amino acids. So each amino acid has a carboxyl group, which is the functional group of a carboxylic acid, and it can donate its proton. It also has an amino group. Amino groups have a lone pair of electrons and can accept a proton, which makes it a base. And so each amino acid can act as an acid and a base. You should be able to identify the N and C terminal amino acids in a protein or peptide. You should be able to describe the primary, secondary, tertiary, and quaternary structures of proteins. Your primary sequence is your sequence of amino acids. Your secondary sequence is the local folding patterns of your protein due to local hydrogen bonding interactions. And those secondary structures are going to be alpha helices and beta sheets. Your tertiary structure is the overall conformation that the protein folds in. And your quaternary structure is when you have two or more peptides or protein strands bound together in a complex. Okay. We are going to talk about alpha keratin and collagen, which are two fibrous proteins. We're also going to talk about hemoglobin and myoglobin, which are two globular proteins. You should know the products of protein hydrolysis. So that amide bond in the protein can undergo hydration reaction or hydrolysis in acid or base solutions, and you will get your amino acids back. You should know what it means when a protein is denatured, okay? And so denaturization destroys your secondary, tertiary, and quaternary structures. It has no effect on your primary structure. We're going to talk about enzymes and how they function. Okay, they have an active site, and substrate and some cofactors may bind to the active site of an enzyme, and catalysis occurs. And then you're going to learn a little bit about how enzymes are used to diagnose certain diseases. Okay, so proteins account for about 50% of the dry weight of our bodies, and unlike lipids and carbohydrates, they are not stored, so we need to eat them every day. The recommended daily intake for protein is 0.8 grams for adult per kilogram of body weight, and more is needed for children. So we can get protein content from our food in meat, fish, beans, and nuts. And protein is made up of amino acids, and they are all the amino acids when it forms a protein. So the amino acids are the monomers, and when you have a protein, those amino acids form a polymer. Okay, so as I said, each amino acid has a C alpha carbon. Alpha is just another way of saying carbon number 2. Um, so this usually when we're counting, the carboxyl group gets priority. So this will be carbon 1, and then the first carbon after the carboxyl group would be the alpha carbon. The second would be beta, the third would be gamma. Okay, so alpha is just a way to number your carbon chain. Okay, so each alpha carbon is attached to a carboxyl group. If you donate that proton, it has a negative charge. We call it a carboxylate group. Okay, it's also attached to an amino group. When that amino group accepts a proton, it has a positive charge, and we call that the ammonium group. Okay, it's also attached to a hydrogen, and then your R group. So there are 20 different R groups for the 20 different amino acids. And each amino acid has a name, but we also can give it a 3-letter abbreviation and a 1-letter abbreviation. Glycine is the simplest amino acid. Okay, simplest because its R group is just H. It is the only non-chiral amino acid because the central carbon is bound to 2 of the same atoms. Chiral meaning that your carbon has to be bound to 4 different groups. Okay, so glycine is the simplest. It's not chiral. The name is glycine. If you want to write the 3-letter abbreviation, it's GLY. The 1-letter abbreviation is G. Okay, so these are the 20 amino acids. Amino acids are generally categorized by the type of amino acid they are, whether they're nonpolar, polar, polar acidic, and then polar basic. Okay, so this chart is off of Pearson, but I like it better than the chart that your book uses. And so up here is all your nonpolar amino acids. Nonpolar meaning they're going to take the R groups are going to contain C and H bonds. There is one exception for tryptophan here, but this is nonpolar. And then there's one exception for methionine. It has a sulfur, but it's still a nonpolar molecule. Okay, so glycine just has an H. Its 3-letter abbreviation is GLY. Its 1-letter abbreviation is G. Alanine is the second simplest amino acid. It has a CH3 group. Valine, if you look at the structure, has 3 carbon atoms attached to it, and it looks like the shape of a V. So it makes it a little bit easier to remember. So a CH and then CH3 and CH3. Its 3-letter abbreviation is VAL, and 1-letter abbreviation is V. Leucine looks almost like valine, but it has that extra carbon group here, so CH2 group here. And then isoleucine looks like an L, so you gotta try to not to get confused. It's not leucine that looks like an L, it's isoleucine that looks like an L. It looks like a backward L. Okay, but all carbon and hydrogen, so very nonpolar. Phenylalanine is got a benzene ring, or a benzene ring with a CH2. Okay, nonpolar. Methionine has a CH2, and then a sulfur atom with a CH3. Proline, if you think of a P, this structure kind of looks like a P on it. It looks like a backward P, huh? And this forms a cycle. Okay, so that cycle looks like the P, the top portion of the P. Okay, and it's the cycle is from the chain, the R group, connecting with an amino group on the that's attached to the alpha carbon. Okay, so it's got 3 CH2 groups, and then it connects to that amino group. Okay, and tryptophan is the amino acid that they say makes you sleepy in turkey, so that's why you get so sleepy after turkey dinner. Okay, and this tryptophan looks kind of like a turkey here, right? This would be its face with the little corner as the beak, and this is its body. That's how I remember the structure. So it's got a benzene ring and then a 5-membered ring. You have a CH2 that's outside the ring, and then the nitrogen. So I just kind of remember the structure based on it kind of looking like a turkey's body. Okay, then you have your polar neutral amino acids. And so polar is going to be attracted to water, right? Because you're going to have those dipole moments and water is polar. So hydrophilic means water loving. Okay, so these polar neutral amino acids have no charge, but they do have polar bonds, and they can be attracted through water through dipole interactions. And then some of them have hydrogen bonding too. Okay, so serine has a CH, our group has a CH2OH. Threonine, think of 3 atoms instead of 2. Serine has 2 atoms and it has an OH. Threonine has 3 atoms with the OH, 1, 2, 3. That's how I remember it. Tyrosine is a little bit harder to remember. It's got that benzene ring and then an OH group. It also has a CH2 group. So it's like phenylalanine with an OH, tyrosine. Okay, cysteine has a CH2SH group, so this one is polar. Remember, methionine is nonpolar. It has that S and then a CH3 group. Asparagine has a CH2 and then it has that amide functional group. And then glutamine has 2 CH2s and then that same amide functional group. Okay, so for this polar neutral, asparagine and glutamine is kind of similar to aspartate and glutamate. Okay, the difference is asparagine and glutamine are nonpolar neutral amino acids. But the A comes before G. The A has 1 CH2 group, the G has 2 CH2 groups. And that pattern is replicated in aspartate and glutamate. Aspartate has 1 CH2 and glutamate has 2 CH2s. It just helps me remember the structure. Okay, and these are polar acidic amino acids, so you have that carboxylate group, okay, or that carboxylic acid functional group at the end of the chain. Okay, so these are polar acidic amino acids. They can donate their proton in solution. It's not shown here at the end. And then you have your polar basic amino acids, and it's histidine, lysine, and arginine. Okay, and basic because you have a nitrogen here that has a lone pair of electrons that can accept a proton. And all of them are shown in their positive charged form. So histidine, and then lysine, and then arginine. These are probably the hardest structures to remember. Okay, I'm not going to ask you to memorize the structure of all 20 amino acids, but you do need to know the names and whether they're polar and nonpolar, acidic, and basic. You do need to know the 3-letter abbreviations and the 1-letter abbreviations. Okay, so I kind of went through the classification on the last table, but we're going to break it down and talk about it a little bit more individually. So amino acids are classified. There's 20 of them. They're classified as nonpolar, which are going to be hydrophobic. There's no polar bonds. They're all nonpolar. And so they will not be soluble in water. And so hydrophobic means water-fearing. If you're not soluble in water, you're going to be a hydrophobic molecule. And so most of the bonds are going to be hydrocarbon bonds, CH bonds. You do have some there that are still nonpolar, but they contain a heteroatom, such as methionine and tryptophan. Okay, then the other classification is polar amino acids. And there's 3 within this classification. You have your polar neutral amino acids, polar acidic amino acids, and then your polar basic amino acids. Okay, and so whether they're classified as nonpolar, polar, polar acidic, polar basic, polar neutral, that determines the properties of the amino acids in aqueous solutions. Remember, these are in your body. Your body has blood in it, which is an aqueous environment. And so depending whether your amino acids are polar or nonpolar determines how it will function or how it will behave in your blood. Okay, so one nonpolar amino acid is valine, just your CH bonds, and then a polar one is asparagine. Okay, this is not charged, it doesn't function as a base, and it doesn't function as an acid. So when you have that amide bond as a functional group in the R group, in your R chain, your N here, even though it does have a lone pair of electrons, is not considered basic. Okay, so this lists all your nonpolar amino acids. Okay, so you have H as an R group, CH3, you have your valine, your leucine, your isoleucine, your phenylalanine, methionine, proline, and tryptophan. So those are all your nonpolar amino acids. Again, you should know, I'm not going to make you memorize all of the structures because your tests are multiple choice, but you should know which amino acids are nonpolar. Okay, so for polar amino acids, we have three different types. We have polar neutral amino acids where your R group does not have a carboxylic acid functional group or an amine functional group to become ionized. Your polar acidic amino acids contain a carboxylic acid group in the R chain, and so a carboxylic acid group looks like that. And again, this hydrogen can come off, and when you get rid of that hydrogen, you form the carboxylate ion. Okay, and a polar basic amino acid, your R group contains an amino group. Okay, so you have a nitrogen on there. Okay, your nitrogen has a lone pair of electrons that can accept a proton, and so when it accepts a proton, okay, you will form a positive cation. Okay, and so that would make it basic. So basic polar amino acids form positive cations, and acidic amino acids form negative anions. Okay, so we have two acidic amino acids. We have aspartate and glutamate. Okay, and so the difference between the two is aspartic acid has a CH2 group with a carboxylic acid functional group, and glutamic acid has two CH2 groups and then your carboxylic acid on your R chain. Okay, so we have three basic amino acids. We have arginine, histidine, and lysine. Okay, so your R group is shown here, and arginine has three nitrogens in its R group. One of them will form a positive charge. Okay, and histidine, the R group is shown here, so it has a 5-membered ring, and this nitrogen can accept a proton and form a positive cation. And then for lysine, we have 4 CH2 groups with a nitrogen at the end. That nitrogen can accept a proton and form the cation. Okay, so there are 20 naturally occurring amino acids. We can only synthesize 10 of them in our bodies. And so the 10 that we cannot synthesize are called essential amino acids, and we must be able to obtain those 10 essential amino acids from our diet on a regular basis. And so the essential amino acids include valine, leucine, isoleucine, phenylalanine, tryptophan, lysine, arginine, histidine, methionine, and threonine. So if you eat meat, meat contains all of the amino acids that you need to eat. If you don't eat animal products, then you have to make sure that your diet is balanced so that you get all 20 amino acids in your diet. Okay, so animal products such as eggs, milk, meat, and fish all contain all of the essential amino acids. But if you don't eat animal products and you're a vegetarian, you only eat plants, then grains, beans, and nuts are deficient in one or more essential amino acids. However, if you eat beans and rice, then the combination of those two will give you all of the essential amino acids. Will give you all of the amino acids, including the essential amino acids. Okay, so how do we draw an amino acid? You always start at the N-terminus, and the N-terminus is going to be on the left side of the amino acid. So you start with the N-terminus, and then you draw your C alpha, and then your CO, C double bonded O, O minus. Sometimes the R group is shown up and the H down. Sometimes the H is shown up and the R is shown down. It doesn't really matter. However you want to figure out how to draw it, it's fine. Okay, so you always draw your amino terminus first, then your C alpha, and then you draw your CO. And I generally draw that backbone first, and then I fill in all my R groups. Okay, so you should be able to look at an amino acid and determine if it's polar or nonpolar. This is our N, C alpha, CO. So this is C alpha, that means it has an H, and its R group is just H. This is lysine, and this is a nonpolar amino acid. Okay. B, here we have an N, C alpha, CO. Okay, so H is not the R group. The R group is shown here. And it's got the CH, CH3, and OH, and that's threonine. Okay, and so this is polar but neutral. Okay, and so this is a polar neutral amino acid. I will say that your amino acids have an N-terminus and a carboxyl terminus. And the N-terminus can accept a proton and become an ammonium ion, and your carboxyl terminus can lose a proton and form an anion. And so you have these two ends of your amino acid that can ionize. And at pH, at the body's pH, which is around 7.2, both ends will be charged. Okay, so you don't, when the amino acid is in your body, it does not have the H on the carboxylic acid group, and it does have the H on the ammonium end, on the amine end. Okay, so here we have an amino acid shown in the picture, and this is the NC alpha CO. Okay, and we have our N terminus and a C terminus. So at different pHs, your acid and base could accept or donate a proton. And at very low pHs, everything that can be protonated will be protonated. And so you'll have an H connected to the carboxylic acid, and you'll have an H connected to the amine. Okay, and that will give your amino acid a positive charge. Okay, once you donate the carboxylic acid proton, okay, then you become neutrally charged, because this is minus, this is plus, and they cancel each other out. And then once the amine gets rid of its proton, then you lose that plus charge, and this is still minus, and so you have no charge here and a minus here, and your amino acid has an overall negative charge. Okay, so if there's three charges that each amino acid can have, it can have a plus charge, it can have no charge, and it can have a minus charge. And those charges will exist at different pHs. Okay, you're going to have a plus charge at very low pH. Around neutral pH, you're going to have no charge, and at very high pH, you're going to have a minus charge. Okay, and so the isoelectric point is the pH. Is the pH when the overall charge of the amino acid is 0. Okay, so that means this is going to be plus and this is going to be minus, and they cancel each other out, so the overall charge is 0. Okay, so for nonpolar or polar neutral amino acids, this is when the Zwitter ion forms. And what is the Zwitter ion? It's a neutral molecule that has both a positive and negative electric charge. Okay, so this is a Zwitter ion. It has a positive and it has a negative charge, but overall the charge is 0 because the positive and negative cancels each other out. Okay, and this is glycine. Glycine has, this is the Zwitter ion of glycine, and its isoelectronic point is at pH 6. So at pH 6, the N-terminus is going to be positive, the C-terminus is going to be negative, and it has an overall charge of 0. Okay, so since amino acids contain an acid, this carboxylic acid group, and an amine, which can function as a base, the carboxylic acid can donate its proton to the base, and so it acts as an acid and a base to itself. Okay, and when that happens, you form your Zwitter ion. So the hydrogen that comes from the carboxylic acid goes to the nitrogen, which functions as a base, and then you form your Zwitter ion. Okay, and that forms a salt. Okay, we know when we said a salt was a metal and a nonmetal, and your metal will be positively charged and your nonmetal will be negatively charged, and so they come together and form a salt or an ionic compound. Okay, this one's a little bit different because it's an internal salt. So this salt is the neutral form of the amino acid, but it has a positive and a negative, and those two together will form a salt. Okay, so the isoelectric point, or the pi, is the pH at which the charged groups on the amino acid are balanced. So you have a minus end and you have a positive end, and so you have an overall neutral compound. Okay, when the pH is lower than the pi, then the carboxylate anion gains a proton and the amino acid has a net positive charge. Okay, when the pH is higher than the pi, than the isoelectric point, the ammonium cation loses a proton, and then the amino acid has an overall net negative charge. And so that's shown in this picture. Here you have your Zwitter ion. It has the plus charge on the N terminus and the negative charge on the C terminus. And this is going to be around a neutral pH. Okay? So if you go to a more acidic environment, that's showing you on this arrow, everything that can be protonated will be protonated. Okay? And so this end is already protonated, but this one's not. So if you go more acidic, there's so many protons floating around that everything that can be protonated will be protonated, and so you're going to add your H2 or carboxylic acid group. And this gives your overall amino acid a positive charge. Okay, as you go up in as you reduce the pH and go up in pH, okay, you're going to start decreasing the number of protons that are floating around in the solution. And so the first thing that's going to give up the proton is the acid, right? So as you go higher up in pH from the positive charged species, you're going to first lose this H on the carboxylic acid portion, okay? And that's when you get your Zwitter ion. And as you decrease the number of protons in solution still and go higher in pH, then you'll finally lose the hydrogen that's on the N terminus, okay? And at that point, your overall charge of your amino acid is going to be negative. Okay, so at neutral pH, you have your Zwitter ion. At very low pH, you have a positively charged amino acid. And at very high pH, you're going to have a negatively charged amino acid. Okay, this says consider the amino acid leucine with a pi of 6. And I'm going to, this is a picture, I believe, out of your book. I'm going to alter this amino acid because the original amino acid that's here is valine. Leucine has another CH2 there. Okay, but it says consider the amino acid leucine with a pi of 6. At a pH of 3, how does leucine change? And so I'm going to draw, this is my C alpha, this is my NH3, we'll draw 3's over here. This is our COO minus, and actually I'm going to draw out that carboxylic acid group. Okay, this is my H. And then I have CH2, and then CH, and CH3, and CH3. Okay, and as I go lower in pH, right, I'm going to add more protons to the solution. Anything that can be protonated will be protonated, and so the nitrogen still stays protonated. This is our C alpha, this is H, this is CH2, CH, CH3, CH3, okay? There's nothing in there nothing in the R group that can be ionized, but now this is going to be let's fix that. This is my carboxylic acid group. And so the overall charge of the amino acid at low pH, around pH 3, is going to be positive. If I go to higher pH, okay, I'm going to be decreasing my hydrogen ion or proton ion concentration, increasing my hydroxide ion concentration. Hydroxide ions will react with any protons that are floating around. And so as I go higher in pH, anything that can be deprotonated will be deprotonated. Okay, and so this is my C alpha. The carboxylate group lost its hydrogen already, so it's going to stay deprotonated. CH2, CH, CH3, CH3. And now I'm going to lose that proton from the base. Okay, so now I'm only going to have 2 hydrogens. Remember, nitrogen likes to have 3 bonds. Okay, so it has 1 to the carbon and 2 to the hydrogen, but now it's not going to have any charge on that nitrogen. And so the overall charge for the amino acid will be negative. Okay, here at the Zwitter ion, or the isoelectric point of 6, it's neutral. Okay, so we're moving on to the different levels of structure for a protein. And the first one that we're going to talk about is a primary structure. Your primary structure is the order of amino acids in your protein. So it's the sequence of amino acids in your protein. That sequence determines how your protein folds and therefore its function. Okay, and amino acids, here's one amino acid, here's another amino acid. They come together in a condensation reaction, and so water will come off and you form this amide bond. Okay, so there's 2 hydrogens on the N-terminus of one amino acid, and the oxygen on the carboxylate end of the other amino acid is used to form water. Okay, so a peptide bond is an amide bond that forms when the carboxylate group of one amino acid reacts with the ammonium ion group of the other amino acid. Okay, and when you have 2 amino acids that join together, it's called a dipeptide. When you have 3 amino acids, it's called a tripeptide. If you have 4 amino acids, it's called a tetrapeptide. Okay, and again, here you have that C double bonded O, and then it's attached to the nitrogen on the next amino acid. This right here is called your peptide bond. Okay, I like this picture much better, and it shows the carboxylic acid group and the amino terminus of the 2 amino acids when they're coming together, and it might be a little bit easier to see how water comes off. But we have 1 hydrogen on the amino terminus and the OH group on the carboxylate terminus, or carboxylic acid terminus, that comes together and forms water. And then you have the carbon of the carbonyl attached to the nitrogen of the next amino acid to form that amide bond. Okay, so this is an amide bond or a peptide bond. Well, it's an amide functional group, but you want to call it a peptide bond. Okay, and that connects the 2 amino acids together. And it is a condensation reaction. Okay, so polypeptides have many amino acids, and your book has a definition of what a protein is. A protein has to have more than 40 to 50 amino acids. And according to this definition, it has to have some biological activity. I also wanted to point out that you can count the number of amino acids in your protein. You want to first find the amino terminus. So start at the left, and then I would start where the first nitrogen is. Okay, and that would be, this is your NC alpha CO. So that would be one amino acid, one complete amino acid. And then NC alpha and then CO is your second complete amino acid. NC alpha CO is your third amino acid. NC alpha CO is your 4th amino acid. NC alpha CO is your 5th amino acid. And then you have the beginning of one and then the end of the other. So that would be the next one. Okay, so when you're looking at a protein, you're always, you should always see the N-terminus on the left and the carboxyl terminus or carboxylate terminus on the right. Okay, and when you draw your protein, you should always start with the N-terminus on the left. Okay, so the amino acid with the free N-terminal group is the N-terminal amino acid. So this is the N-terminal amino acid here, and this is the C-terminal amino acid. Okay, and so it's always written like that. N-terminus on the left, your C-terminus on the right. When you're naming proteins, you're going to name the protein, the last name of the protein is going to be whatever amino acid is on the C-terminus. Okay, so that C-terminus amino acid will be the final name in the protein, and all the other amino acids will be named as substituents. Okay, and so you're going to put the name of the amino acid with the "il" ending. Okay, and you're going to prefix all of those names to that C terminal amino acid. So alanoserine is the name of this dipeptide. Okay, so some example of biologically active peptides are enkephalins. So enkephalins are pentapeptides, so they're proteins made up of 5 amino acids. They are made in the brain and they act as painkillers and sedatives. And the way they work is they bind to pain receptors. And so if you, I think I only have one of the pictures here. Okay, so this is NC alpha, NC alpha CO, so that's 1 amino acid, NC alpha CO, that's 2 amino acids. And let's just go through here. So that's 1 amino acid, that's the 2nd amino acid, this is the 3rd amino acid, this is the 4th amino acid, and that's the 5th amino acid. This is tyrosine. Okay, the next one just has an H, so that's glycine. Okay, the next amino acid just has an H also, so that's still glycine. Okay, and the next amino acid has a CH2 with a benzene ring, that's phenylalanine. Okay, and the last amino acid, a CH2, and that would be leucine. Okay, so the name will be tyrosyl, glycyl, phenylalanine, leucine. Sorry, okay, and that's yeah, so that's shown here in the 3-letter abbreviation. All right, so these are 5 amino acid proteins that act as painkillers in your brains. They're called enkephalins. And morphine and heroin work by binding to the same receptors that these 5 pentapeptides also bind to. And so they produce similar physiological effects, but their effects are longer lasting. Morphine and heroin have longer lasting effects. So enkephalins are related to a group of polypeptides called endorphins, which are known to reduce pain, and they're supposed to enhance your mood. Endorphins are what you generate when you exercise a lot. Okay, so proteins, anything that has more than 40 to 50 amino acid can be considered a protein. Everything below that is a peptide. Okay, and there's lots of different proteins, and these different all these different proteins can have different characteristics and different functions. So some proteins are structural components, such as cartilage, muscle, hair, and nails. Other proteins function as enzymes and regulate biological reactions and metabolism and digestion and cellular metabolism. And then some proteins do other functions, such as transport oxygen in the blood, such as hemoglobin and myoglobin. So there's not one function of a protein. There's lots of different kinds of proteins, and those proteins can have different functions. Okay, and some of the proteins that we're going to talk about is hemoglobin. Hemoglobin is responsible for transporting oxygen in your blood. Collagen is a fibrous protein. It's found in connective tissues. It's found in your skin, tendons, bones, and blood vessels. Ferritin is a protein that stores iron in the liver. Actin and myosin are proteins that help your muscles contract. Okay. Keratin is a fibrous protein found in hair and nails and skin. Myoglobin helps store oxygen in tissues. And insulin is a protein that helps control blood sugar levels in your body. So there's lots of different types of proteins, and they have lots of different functions. Okay, so looking at the different levels of structure for protein, we've mentioned the primary structure, which is your sequence of amino acids. It's a sequence of amino acids in your peptide or protein. and that sequence will determine how that protein folds. Your secondary structure is a conformation assumed by the protein's backbone, okay? And so it's the local hydrogen bonding interactions that create these secondary structures. And the two secondary structures are alpha helices and beta sheets. An alpha helix looks just like it says, it looks like a helix, okay? And it's when the hydrogen on a nitrogen, hydrogen bonds to the lone pair of electrons on an oxygen on a nearby carbonyl group, okay? And these hydrogen bonds are generally around 3 to 4 amino acid residues apart, okay? The other type of structure that you can have is this beta sheet, beta pleated sheet, and so the structures for the alpha helix and beta sheet is a little bit different. For the alpha helix, those hydrogen bonds are in the same chain and they wrap around because those hydrogen bonds are around 3 to 4 residues apart. Okay, for beta sheet, your chain for your amino acid is going to come back and then wrap around on itself. Okay, and so those hydrogen bonds are going to be more amino acids apart. Okay, and it's more of a side-by-side interaction. Those polypeptide chains or protein chains are going to wrap around and form this zigzag pattern, and then there's hydrogen bonding between the side chains of the polypeptide. Okay. Okay, the tertiary structure that we're going to talk about is the overall three-dimensional structure of the entire protein. And that has to do with intermolecular forces. So secondary structure is mainly local hydrogen bonding interactions between the polypeptide chain. In tertiary structures, we're going to have all different types of intermolecular forces like hydrophobic interactions, hydrophilic interactions, salt bridges, which is ionic interactions. You're also going to have hydrogen bonds, and then you're going to have disulfide bonds. So you have all these intermolecular forces at play to hold your protein in a specific 3D conformation. And then for quaternary structure, it's when you have two or more individual peptide chains come together to form a complex. And so this is hemoglobin, and hemoglobin will have 4 different chains. So it has 2 A chains, so that's shown in red, okay, and then 2 B chains, which is shown in orange. And so these are 4 polypeptides that are individually and distinct, but they bind together to form this complex. Okay, and you can see the alpha helices here. Okay, so this is secondary structure. You can see all of the different alpha helices here, and I don't see any beta sheets in hemoglobin. Okay, all right, but we will see some more. Okay, so like I said, the primary structure of the protein is a sequence of amino acids joined together through peptide bonds. And all the bond angles are 109.5. So your carbons that are, this is nitrogen, this is carbon, this is the carbon, that's the C alpha carbon, and this is the C double bonded O. That's part of the carboxylate end of the amino acid. So for this carbon, it's going to be tetrahedral. Okay, this carbon has 4 electron domains, 1, 2, 3, 4, and so that gives you tetrahedral geometry, and the bond angles for tetrahedral geometry is 109.5. This carbon has a double bond to oxygen, and then 1 bond to the C alpha carbon, and then to nitrogen. So this carbon has 1, 2, 3 electron domains. 3 electron domains gives you trigonal planar geometry, and so the bond angles for that carbon is 120 degrees. Okay, so you have tetrahedral and then trigonal planar. This nitrogen here, it has a lone pair of electrons that are not shown. So if you're counting electron domains, you have to consider the lone pair of electrons. So the lone pair of electrons is 1, 2, 3, 4. 4 electron domains gives you tetrahedral geometry, but we don't see the lone pair, and so this will be trigonal pyramidal geometry. And so we don't give you what the bond angles for that is. It's still going to be around 100. Actually, it's still 109.5. So it's 109.5, this is 120, this is 109.5, 109.5, 120. And so those bond angles will give you this zigzag conformation of your protein. Okay, so we learned earlier how to draw an amino acid. So now we're going to learn how to put all those amino acids together in a protein, okay, or peptide. Okay, so amino acids are linked together through peptide bonds. The carboxylate group on one amino acid will react with the ammonium ammonium end on the next amino acid. You're still going to start at the N terminus. So starting from the left, draw the amine group. So NH3. Then you draw your C alpha group, okay? And then finally you add your carbonyl group from the carboxylate end of the amino acid. Okay, and then you're just going to attach in an amide bond, okay? So the H2 from the nitrogen joins with the oxygen here. You get rid of it with water, okay? And then you form this amide bond in between amino acid 1 and amino acid 2. Okay, and then you have your C alpha and then CO, and you do the same thing over again. Okay, and so the way I draw my peptides or my proteins is I always do the backbone, NC alpha CO, NC alpha CO, NC alpha CO, and then I fill in my H and R groups. Okay, I just find that easier, and I'll show you an example. Okay, so this says draw the dipeptides serine and threonine. And so I'm going to start with that N terminus. Oops. I guess I want to do black. Okay, so that's my N terminus. That's the NC alpha, and that's our CO. And then we have N, and then C alpha, and then CO. Okay, and then I can go and fill in my hydrogens, and then I'll fill in my R groups. So serine just is CH2, and then, whoops, let's clean that up. OH. Remember, carbon has to have 4 bonds. So it has 2 to hydrogen, and then 1 to the C alpha carbon, and 1 to the OH. And then for threonine, it looks almost like serine, but it has 3 atoms. That's a 3. And this is OH, but it has 3 atoms. Okay. And so that would be your dipeptide. Serine becomes seryl, and then threonine stays threonine. So seryl threonine. Okay, this question says, what is the N-terminal amino acid? So this is the N terminus, and this is the C terminus, and this is the N-C-alpha-CO. That's the first amino acid. N-C-alpha-CO is the second amino acid. N-C-alpha-CO is the third amino acid. This one is phenylalanine. This one is cysteine. And the last one is just alanine. So the name will be phenylalanyl cysteine. I probably spelled that wrong. Alanine. And they're all kind of connected. Okay, this says write the names and 3-letter abbreviations of tripeptides that could form from 2 glycines. So we have glycine, glycine, and alanine. So that's one. And we've got to come up with, write the names and 3-letter abbreviations of, okay. So that's one. Another would be glycine, alanine, glycine, oops. And then we would have alanine, glycine, and glycine. So that's 3 different peptides that you can get from the same 3 amino acids. And so this will be glycyl, alanine. This will be glycyl, alanyl, glycine, and this will be alanyl, glycyl, glycine. Okay, so we're going to talk a little bit more in detail about the secondary structure of the proteins. And so your secondary your secondary structure is the 3D arrangement of the localized regions of the protein, and their structured regions arise due to hydrogen bonding between the H on the nitrogen of the amide with the C double bonded O with a lone pair of electrons on the oxygen from the carbonyl group of another. And so we get two stable arrangements. One is called the alpha helix, and the other is the beta pleated sheet. And in the alpha helix, the peptide chain twists into a spiral, and you get hydrogen bonding between the hydrogen that's on the amide group and the lone pair of electrons on the carbonyl group, that are around 3.5 to 4 amino acids apart. And the beta pleated sheet, we have two or more polypeptide chains that line up side by side, and you have hydrogen bonding occurring between the chains that are lined up. Okay, so here we're looking more closely at the secondary structure called the alpha helix. And you can see in the picture, for example, you have the hydrogen on a nitrogen here. That's hydrogen bonding to the lone pair of electrons on an oxygen further up in the chain. Okay, so hydrogen bonds form between the lone pair of electrons on an oxygen that's part of a carbonyl, and the hydrogen that's on the nitrogen that's part of the amide bond in the next turn. Okay, so the formation of many hydrogen bonds along the polypeptide chain gives the helical shape, and it looks like a spiral staircase that's kind of shown here. Okay, all of the R groups, if you look at the R groups, they're shown in purple. They're all sticking on the outside of the helix. Okay, none of them are pointing towards the inside. So all of the R groups extend outside the helix, and they generally, the structures that develop an alpha helix, they usually have large R groups, such as histidine, leucine, and methionine. Okay, so for the beta sheet, instead of the polypeptide chain spiraling on itself and forming hydrogen bonds that are around 3.5 to 4 residues apart between the hydrogen on the amide nitrogen and the lone pair of electrons on an oxygen. And instead, we have the chains running alongside each other in parallel. Okay, and so then you'll form hydrogen bonds between the hydrogen on the amide nitrogen and the lone pair of electrons on the oxygen that's part of the carbonyl group. Okay, and so generally your structures that form beta sheets, those R groups are sticking above and below the plane of the paper. See, the beta sheet looks like a zigzag, and it's in a plane, and it kind of looks like if you had taken a piece of paper and folded it like an accordion. Okay, your R groups are sticking above and below that sheet of paper. So the R groups are purple, and so you can see this purple R group is sticking above the paper, and then this one's sticking above the paper. This purple R group is sticking below the paper, so is this R group sticking below the paper. So above, and then below, and then above, and then below. Okay, and for structures that adopt a beta pleated sheet, generally your R groups on your amino acids are going to be small. So they're going to be glycines, valines, alanines, and serines. Okay, so most proteins have alpha helices and beta pleated sheets, as well as regions that cannot be, that are not well organized or not characterized. Okay, and so we call those kind of loop regions. Any of these, see these little wire stems? Okay, a lot of times those are loop regions or disordered regions. Okay, shorthand symbols, which we call ribbon diagrams, are used to indicate these regions of secondary structure. So they show an alpha helix like a flat ribbon shown in a spiral helix structure, and then beta sheets are shown as flat arrows, and then random loop regions are shown like it's like a wire. Okay, so now we're going to talk about the tertiary structure of the protein, and that's the overall 3D conformation that your protein adopts. And in general, your protein will push all of the nonpolar hydrophobic amino acids towards the interior of the protein and put the more polar or charged amino acids towards the exterior of the protein. Why? Because nonpolar residues will want to be near other nonpolar residues. They'll be hydrophobic or water fearing, whereas the more polar or ionized amino acids will be more soluble in the water and so more stable when it's on the outside of the protein that's exposed to the aqueous environment of the blood. Okay, so to maximize hydrogen bonding with water, the nonpolar side chains are stabilized by London dispersion forces in the interior of the protein, so they get pushed to the inside of the protein. And the polar functional groups with hydrogen that's attached to either an oxygen or a nitrogen that can hydrogen bond are more pushed towards the outside. Also, amino acids with charged side chains are attracted to each other through electrostatic interactions or through polar interactions with water. And then also you have disulfide bonds that form covalent bonds that stabilize the tertiary structure. So the intermolecular forces that we're talking about are London dispersion forces, dipole-dipole interactions, and hydrogen bonding. And disulfide bonds are the only covalent type of interaction that you have in the tertiary structure of your protein. And just to be clear, if we have a disulfide bond occurs when you have two hydrogens, and this would be your C alpha, and that would be the rest of your chain. Okay, CH2, and then this is your C alpha on another polypeptide chain. So these two sulfurs can form a disulfide bond and be covalently attached to each other. So this is a nice picture that shows all of the different types of tertiary interactions or the intermolecular forces that occur in the tertiary structure of your protein. Okay, and these are the interactions that cause your protein to bend and twist, and it's determined by cross-links, the attraction and repulsions between the R groups, different R groups of all the amino acids in your polypeptide. Okay, so you can have hydrogen bonding, which is shown here, which is what we talked about when you have a hydrogen on, let's do black. Okay, you have a hydrogen on a nitrogen, that's hydrogen bonding to the lone pair of electrons on a carbonyl group. A lone pair of electrons on an oxygen that's part of a carbonyl group. It's part of a polypeptide chain. Okay, so that's what those dotted lines represent, hydrogen bonds. Okay, here you have another hydrogen bond. Again, the lone pair of electrons are attracted to the hydrogen that's attached to a very electronegative atom, such as nitrogen. Okay, here we have a London dispersion forces, or what we can call hydrophobic interactions. Those are nonpolar interactions. London dispersion forces occur between nonpolar molecules. So this is valine that's interacting with, that is leucine. Okay, so those are both those amino acids are nonpolar, and so you'll have London dispersion interactions between those two amino acids. Here we have hydrogen bonding between the hydrogen on the nitrogen of asparagine and the lone pair of electrons on the oxygen of a nearby carbonyl group. Here we have a salt bridge. This is an electrostatic interaction, and this is occurring between the positively charged nitrogen on lysine and the negative charge on aspartate. Okay, so that's a salt bridge or electrostatic interactions. Okay, down here we have phenylalanine, and over here we have leucine. Okay, so those are very nonpolar amino acids, and again you have those hydrophobic interactions, or we can call them London dispersion forces. Here we have a disulfide bond between two cysteine residues. And this one's showing hydrogen bonding between the hydrogen on a serine and the lone pair of electrons on a threonine. Okay, and then here we have just hydrogen bonding holding your alpha helix together. Okay, so it shows you all the different types of intermolecular forces that hold your protein in its tertiary structure. This is a nice picture where you have to describe what the intermolecular forces are that are holding your protein together. And so for this one down here, we have hydrogen bonding between a aspartate and a water molecule. So that's just hydrogen bonding. I hope I can write small enough here. In D, this region here, okay, this is isoleucine and 2 isoleucine residues, okay, and so that is forming an intermolecular interaction between the two with hydrophobic interactions. Hydrophobic, I can't spell. And, or you can call that London dispersion forces. Okay, so those are no non-polar interactions. Here we have two, it looks like a 5, it's supposed to be an S. So that's a disulfide bond between 2 cysteine residues. Here we have also more London dispersion forces. Or what we can call hydrophobic interactions. Okay, here we have a salt bridge that's happening between carboxylic acid, that's the C-terminus end of one amino acid or protein, and lysine, okay? Or we can call those electrostatic interactions. Okay, so let's go up here. Here's another example of electrostatic interactions or a salt bridge. And that's through the N-terminus of the polypeptide, and that would be glutamic acid. Okay, for this one, this interaction here, okay, that's aspartate is the so the hydrogen on tyrosine is hydrogen bonding to the lone pair of electrons on aspartate. Okay, so that's hydrogen bonding. And then here we have 2 phenylalanines interacting, and that's still hydrophobic interactions. Okay, those are nonpolar amino acids. Or we can call that London dispersion forces. Okay, and then here, well, here we just have, I think this hydrogen is supposed to be hydrogen bonding to the lone pair of electrons on the oxygen. So that's hydrogen bonding. It's just pointed to the C and it should be pointed to the O. And here you also have hydrogen bonding. So you have lots of different types of intermolecular forces holding your protein in its tertiary structures. Those include London dispersion forces or hydrophobic interactions. They are dipole-dipole interactions, which are polar. We call those hydrophilic interactions. And I don't see any here. Maybe this hydrogen bonding would be considered could be considered hydrophilic interactions. And then we have salt bridges or electrostatic interactions, disulfide bonds. Okay. Okay, so written all out, your tertiary structure of your protein is due to 1, hydrophobic interactions, which are also called London dispersion forces. Those are interactions between 2 nonpolar amino acids. Again, nonpolar amino acids want to be away from water, so they get pushed towards the interior of your protein. Hydrophilic interactions, which could be another word for dipole-dipole interactions, are attractions between external aqueous environment, or the attractions between the external aqueous environment and the R groups of polar amino acids, which are pulled to the surface of the protein, which is exposed to the water. Okay, so like dissolves like, or you have similar interactions, and we can call those hydrophilic interactions or dipole-dipole interactions. Okay, then we have salt bridges, which are ionic bonds They're ionic bonds, so they form between a cation and an anion. So if one amino acid side chain has a cation and another has an anion, then they can form a salt bridge, or we can call those electrostatic interactions. Okay. Hydrogen bonds occur between a hydrogen that's on a very electronegative atom, such as oxygen and nitrogen, and the lone pair of electrons on a nearby oxygen or nitrogen of another amino acid. Okay, and then we also have sulfide bonds. So sulfide bonds are the only covalent interaction that's involved in the tertiary structure. Okay, and they form between two sulfur groups that's part of a cysteine on two different amino acids. So this is a study check. Select the type of tertiary interaction. So leucine and valine, those are both nonpolar amino acids, so that's going to be hydrophobic. And maybe I'll just draw a line. Okay, we'll just circle. Okay, two cysteines is what's going to form a disulfide bond. Aspartic acid and lysine. Aspartic acid is the R group has a carboxylic acid group, and lysine has a basic amine. Okay, and so when both of those species are charged, you form a salt bridge or an ionic interaction. Okay, and then serine and threonine both have a hydrogen attached to oxygen, and so you can have hydrogen bonding occurring between those two residues. And so, let's see, we'll just go around here and point it there. Okay, so moving on to the quaternary structure of the protein, it's when you have more than one polypeptide in your complex. Okay, so two or more folded polypeptide chains come together in one complex. One example is insulin here, another is hemoglobin that's shown here. So insulin contains two separate polypeptides shown in blue, and they are joined together through disulfide bonds through the cysteine residues here. Okay, hemoglobin consists of 4 different polypeptide chains. And so here's 1, 2, 3, 4, and they're held together by lots of different intermolecular forces. And it's held in this quaternary, it's a tetramer, so it has 4 different parts held together in a complex. And it uses lots of different intermolecular forces to do that. Okay, so just to review what we said, the primary structure of the protein is just the sequence of amino acids, okay, and that determines how the protein folds. Your secondary structure are your alpha helices and beta sheets. Your tertiary structure is the overall 3D conformation that your protein adopts, okay, and then your quaternary structure is when you have more than one polypeptide form a complex to form the protein. Okay, so we have another study check. Identify the level of protein structure. So the beta pleated sheet is going to be secondary structure. So that's secondary. The order of amino acids in a protein is the primary structure. C, a protein with 2 or more polypeptide chains is quaternary structure. D, the shape of a globular protein is tertiary structure. And disulfide bonds formed between R groups is still tertiary structure. Okay, so we're going to finish the lecture off with our active learning worksheet and continue with lecture for the part 2 of chapter 16. Okay, and it says protein contain 4 elements. Those are carbon, nitrogen, hydrogen, and oxygen. And they sometimes also contain sulfur, depending on the particular protein. Proteins are polymers consisting of long chains of amino acids. There are 21 naturally occurring amino acids, so they've added one over the years, which all have the same basic structure. They all have an amine group and a carboxyl group. However, the blank or R group is what makes the amino acids different from one another. So it's the side chains, or the R group makes them different. Then it says draw the molecular structure of an amino acid below. draw a different colored ring around the following and label. Okay, so we're going to have the amino terminus, the C alpha, and the carboxylate terminus. Okay, then we're going to have an H, and then we're going to have an R group. Okay, and we can make the acid red and the basic blue, and I forgot to put the charge there. Oh, it's turned color here. Okay, and so draw a different colored ring around the following and label the amino group. This is the N terminus, this is the C terminus, and this is the R group side chain. That's fine. And it says glycine is the simplest amino acid with just a hydrogen atom as its R group. Draw the structure of glycine. So this is NH3, this is your C alpha, and that's your CO minus, and H, and then the R group is H. Amino acids are buffers. If dissolved in water, both the amino and the carboxyl group of an amino acid can ionize. The carboxyl group, as seen in a fatty acid 2, can ionize into a proton and a carboxylate group if the proton is what gives the molecule its acidic properties. However, the amino group can act as a base and it can accept a proton or hydrogen ions, changing from NH2 to NH3+. Amino acids are described as being amphoteric due to the fact that they have properties of both an acid, releasing protons in solution, and a base, accepting proton or hydrogen ions in solution. This also means they can act as buffers, helping to regulate pH inside an organism. Okay, and then the next column says formation of a dipeptide. Okay, and so the name of the reaction going in the forward direction is condensation, and going in the opposite direction, it's hydrolysis. This could also be called dehydration, synthesis. Okay, and so what you get is H and an OH coming off to form water, and then in this form, an H2C. Here they wrote the H down and the R up, which is fine. You have your C double bond O, and then your nitrogen for the amide bond, and then your C and your R and your H, and then your COOH. So here they have it in the non-charged form, which is not what occurs at regular pH. Okay, so we drew the dipeptide, and this bond formed is called a peptide bond. Okay, functions of proteins. List the ways proteins play a role in an organism's body. Give specific names of examples if you want. Okay, so the different types of proteins that we have could be structural proteins, and that's proteins like collagen and keratin. Then we have transport proteins, sorry, I can't spell, such as hemoglobin and myoglobin, and they transport oxygen. Hemoglobin transports oxygen in the blood to all over the body, and myoglobin helps transport oxygen in tissues and store oxygen in tissues. Then you have contractile proteins. Oops, maybe I should rewrite that. Contractile proteins. And actin and myosin are two examples, and they are in muscle. Okay, and then we have enzymes that form catalysis in the body and are involved in reactions in metabolism and cellular respiration. So and there's a lot of them. If I can think of one that I'm interested in, and I was trying to get you guys to do a research project on, but we went in quarantine instead, is malate dehydrogenase. Okay, so that's good enough. All right. Okay, so that brings us to the end of part 1 for chapter 16. And what did we learn so far? We learned the structure of amino acids. Okay, you have your C alpha carbon that's attached to the amino group, and then a carboxylate group. You also are attached to a hydrogen and then an R group, and each amino acid has a different R group. There's 20, 21 amino acids. We learned that your amino acid can function as an acid and a base. You should be able to look at a peptide and label the N terminus and the C terminus, and then label those amino acids. You should know the different levels of structure for proteins, so primary all the way to quaternary. We didn't talk about it, but this is what we're going to talk about next time. We're going to talk about alpha keratin and collagen, hemoglobin and myoglobin. We're going to talk about the products of protein hydrolysis and protein denaturization. And then we're going to discuss enzymes and how we use enzymes as a way to diagnose disease.